Perfect, but we're basically at time, so we'll move into the second section of the day, which the next presentation is the next generation tools and cancer pipelines. You heard from the team yesterday about the next gen tools, and now we'll build on that, focusing exclusively on the tools that have been developed for cancer research. So I'm very happy to introduce Daniel Marrama to the stage to present next gen tools and cancer pipelines. Thank you, Dan.
Thanks, Nina. Hi, everyone. So yeah, I'm Dan. I'm a bioinformatics specialist on both the IEDB and CEDAR teams. And yeah, so I'll be going over some of the cancer-specific tools that we use in cancer pipelines that you could also use for your workflows on the NextGen site. So as an outline for this presentation, I'm going to just show how we can access those tools and run this specific example of a new epitope prediction pipeline using these three tools together. And you'll see how we can easily pipe those tools together on the NextGen site. And then I'll go into more details on how advanced users can use these tools outside of the NextGen website or the browser, both as standalone versions or using our API. And I'll go over some other info as well. So before I do that, I'll just give some basic context on neoepitopes. So they are peptides that arise from mutations in cancer cells. So when a mutation occurs inside of a cell that leads to a cancer, like let's say a RAS gene, which is those genes are responsible for cell growth and division, and can lead to a cancer. Then downstream from those mutations, you can have the protein product itself is mutated. And then when those are chopped up and presented on MHC binding proteins that are just a normal process for all cells, these what we call neoepitopes can be recognized by a T cell that has gone through the negative selection process so that they can be um, used, and this is how your immune system can keep cancer cells in check in your body. So to access these tools, you've been familiar with if you were in day two yesterday, you're familiar with the legacy and next gen tools. So on the CEDAR homepage, in the analysis resource tab here, there's the next gen tools link and this legacy link as well. So these are all the tools on the legacy site, and some of these we had gone over yesterday. And so if I go back to the CEDAR homepage and click the next gen tools site, it leads me to this page, and here are all the tools that you can use for specific pipelines. And then also on the CEDAR homepage, on the right-hand side, these are the cancer-specific tools that you can click that lead you directly to the NextGen site automatically. So this is the first tool we're going to go over, the mutated peptide generator. So as I explained from these mutations, this is basically generating peptides that could potentially arise from those mutations. And so there are two main categories of mutations that we consider. In-frame mutations include we can have like a single nucleotide variant or a polymorphism, otherwise known as a SNP, where it's just one single nucleotide that's changed or mutated. And in this case, you're going from this one nucleotide can change a phenylalanine to an isoleucine, and that can be again like recognized by the immune system in a sequence as a new epitope. And you can also have insertions or deletions that keep the open reading frame the same as normal. So it would just be adding or deleting a downstream amino acid. So the other category I mentioned is frame shift. So this would be insertions or deletions that would shift the entire open reading frame itself. And so all these downstream amino acids can change into totally different ones from the wild type. And so again, these can arise as neoepitopes when presented to T cells. So the tool itself uses what's called a VCF file or variant call format, and these will be compared to some reference. So this standard chromosome number, position, and reference and alternative nucleotide will be these are comparisons to like a reference, like I said, and from that we can annotate these variants that will identify transcripts that may be impacted by this mutation, and then downstream from that, you can generate protein or sorry, peptide sequences that are affected. So in this case, this normal sequence, which has a glutamic acid here, can be mutated, and aspartic acid is what is shown up downstream after this mutation happens. So for the accessing this mutated peptide generator, you can click on it here at the bottom, and you can also so first you can upload your VCF file by clicking this button here, or you can also drag this file your file into this box. And so if we scroll down a little bit, you can see here the standard headers for the VCF file. So this chromosome number, position, ID, alternative, and reference nucleotides. And so these are also here at the parameters that you can select. So I'm going to select peptide length as 9, the mutated position as 5, and then all the overlapping downstream frameshift will be 8, and the maximum peptide length I want is 10. And so you can also run this SNP-F annotation, which it uses, but since this VCF file already contains those annotations, I can skip that. But you should check the box if your VCF file does not have that. So when I click run, I will get a table that we can use for more downstream pipelines to the next tool. So this table, I'm just going to shortly shorten it so I can show you that over here on the right-hand side, the 2 columns here is the reference peptide. So this is from the wild type sequence, and the mutated peptides that are generated using this tool. So this is a threonine to an alanine, or sorry, an alanine to a threonine, I should say. And then, so the next tool I'm going to use to pipeline those results to the next to the next tool in our pipeline, we're going to use this peptide variant comparison tool. So this takes 2 lists of peptides together and does a comparison on MHC binding or immunogenicity prediction scores. So in the context of MHC binding, as you may know, then in this MHC molecule here can bind to this orange peptide here, and there are these specific anchor mutations that are more important than these other positions that are involved in the binding affinity. So this specific HLA-C 1502 allele, I can show this motif plot, which shows the enrichment of specific amino acids at certain positions. So the serine, alanine, threonine are very important in this first position here, and these leucine, valine, and isoleucines are very important at this position here. So we can compare the 2 lists that we just generated, the reference and the mutated peptides together to see how binding is affected. And then we can also use this isoFire immunogenicity prediction method, which is an ensemble of random forest machine learning models that takes the MHC binding features themselves along with some other core like the core of the peptide itself and downstream features, as well as peptide abundance from TCGA that improves immunogenicity predictions, as you can see here. So the peptide variant comparison tool includes both just MHC binding itself and this isoFire method. So when I go back to my results, I can click this plus button and scroll down and click this variant comparison tool and use the unique peptides themselves. So I don't want to use all of them, or just the ones that are unique, and so there's no redundancy. So it automatically selects the 2 columns that we generated. And you can see that this, just by clicking that tool, we have this second step in our pipeline compared to our first step here. So it nicely on the browser generates this second step. And then here you can specify the prediction models that you want. So here we have MHC, net MHC pan 4.1, as well as the immunogenicity predictions, isoFire. I'm going to remove net MHC pan because isoFire includes that as part of its prediction. Um, and so up here as well, you can specify specific alleles. So if you're looking at a specific patient and you want to know how mutated peptides might bind to their specific alleles, you can add those in here. And then we can just click run, and it will take those same two lists of peptides from those columns and generate these immunogenicity prediction scores. And when I scroll down here, you can also see the two columns, peptide A, which is the reference, and peptide B, which is the mutated peptides that we generated. You can also display other columns if you want. So by default, we're just showing some of the columns from IsoFire, but you can also show the specific MHC binding EL rank itself, let's say for the mutated peptides. So if I click that and click display, that will show up as a separate column here. And then I can also do additional filtering on these. So let's say I want to take the top 2% rank of these peptides, and then I can save this as this table state and use that to pipe those specific filtered results to the next tool. So the next tool we'll pipe to is called the Peptide Expression Annotation tool, or the PEPX tool. So this is a tool that takes various expression data from different database sources, and it'll output TPM values, or transcript per million values, and how abundant those peptides are in these specific data sources. So this is useful because it's been shown in the past that expression and MHC binding is correlated in some way, and it improves T cell epitope predictions. So you can do the same thing as we did earlier and click this plus box and go down and click PepX. And I'm going to click the peptide A column because these are the reference peptides. They'll be found more likely found in these data sources. And I can click here TCGA, the Cancer Genome Atlas, and you can click the specific tumor sample or tumor type you want. But I can just click the pan-cancer data set. And from here, I'll get all of the TPM values for those peptides and the genes that they are found in. So you can see at the bottom here, there's this one peptide that has a lot of expression from various different genes. And then I can also download these results into a TSV or CSV or JSON format and use these as in downstream in my analyses as well. And so I want to also show that on the left-hand side, as we generated these tools, you can see if you click these arrows here, all the parameters that we used for this pipeline. And at the top here, I can name this pipeline itself and save it and email it to myself or to a colleague. Let's call it new epitope prediction, and I'll email it. And then that will be sent as the link to some yourself or someone else. And then at the top here, you can click this button and copy it as a URL. And so if I close the tab and input that URL into the top here, I'll get the exact same pipeline that we just ran with all the same parameters that we used, as well as the data that was generated is saved as well in here. So this is nice if you want to replicate workflows from colleagues, or you want to reproduce or rerun things that you've already done before. And next, I'm just going to show that if you click here at the top, the tools bar, and click this tools list, here's all the tools in the next-gen site. And on the left-hand side, there's these different tags. So if I click the cancer tag, these are all the tools specific to cancer. And so next I'm going to show for standalone, sorry, I'm going to rerun this. So if you want to move away from the browser and run these tools on your own machine, you can go to any of these tools and click this download button. And it will bring you to this generic downloads page, which shows all of the different tools that are available as standalone packages that you can. So if I click the mutated peptide generator here and click agree to download, then I will get this zipped up package with the tool itself running in here or inside there, and there'll be some README file as well as all the different code that is needed to run the tool. And then next, if you want to use our API, this is a convenient way to send HTTP requests to our servers. So again, this would be moving away from the front end. And the way this is done is a two-step process. So you can submit a post request to our server with all of the data and parameters themselves. And what you'll get in return is a pipeline ID and a results ID. And the reason we do this is because some of the pipelines and tools can take a while. So you'll get an ID that then you can do another request or submit as a GET request, and it'll give you the status of that job or the results themselves to see if they're available. So the way I do that, I'm going to show how to actually do this in the browser, but you can do this, of course, just in your terminal or in some kind of script. So here, if I click the copy API post request, this will actually generate all of the JSON data that is needed for this to submit this post request. So if I go back to the homepage of the NextGen site and click the API button down here, these are all the API endpoints for the specific tools themselves. There's also this convenient post request API endpoint for pipelines. So I can submit an actual entire pipeline itself with each individual data in there. And so if I try it out here, I can just delete all this in the box and paste that exact same JSON that I copied from earlier. And then I can run this, of course, here within the browser itself, and you can see the first step of the mutated peptide generator here. And the second step is the peptide variant comparison tool here. And then the third step, of course, is the PepX step here. And so when I click execute, I'll get, like I said, well, here it'll actually show all the entire curl string that will submit this post request. And here's the response I'll get in return, which are various information relating to the pipeline. So specifically, the most useful is this result ID, because this is how you'll get the actual results. So it's running in the background on our servers. And if I go down to the results endpoint here, I can do the same thing and submit this result ID within this box. And if I click execute, this is all the data that's been returned from our pipeline, and this is the exact same data that we saw on the front end. And so again, I just want to stress that I'm showing this in a browser for convenience, but you can, of course, just write some scripts that do this post and get requests automatically to automate your workflows very nicely. And also, if you're interested in how the different parameters we used in these pipelines or for other tools, we have extensive documentation. So for the mutated peptide generator itself, there's all these explanations for each of the parameters that we used. And if you want to go even further than that, you can also go to this Cite Us page here at the top right. And these are all the papers that have been published in relation to the different tools. So if you want to go even further in understanding how the tools work, you can read these as well. So I'd just like to reiterate that you can contact us if you have any issues with running your pipelines or workflows at either of these email addresses. And if you're interested in a paid license for non-academic use, you can contact us here. Thank you very much. I'll just pass it back to Nina.
Excellent. Thank you so much, Dan, for that super detailed walkthrough of a few of our really important cancer tools. And for those of you who joined yesterday, hopefully you can see how we've been building on day the materials that we've presented. So a lot of the features we touched on yesterday, and you see how in the next gen tools they come back again. So again, the next gen tools is an example of being able to leverage our existing capabilities that were designed for IEDB, but now can be adapted for the cancer setting. And it's a new platform, so it has the latest technology and infrastructure, and now we can, use it and implement our cancer tools as well. So hopefully between yesterday and today, we've covered a really good amount of the next gen tools, and it gives you the opportunity to have a look and test out some of the tools, some that you may be familiar with based on your experiences with a legacy site, and then some new ones that you can maybe take a look at and apply to your research.